Okay, so now what we're going to do is you're going to get ready to electroporate myxobacteria with the DNA that you isolated from that plasmid-bearing E. coli, okay, which I call it P. mycomar because it's a mycoplasma, something called mariner. So it's in my book as Hymar, but we're saying P. mycomar, but that's fine. So what you're going to be given is a flask with myxobacteria in it, and you're going to, first step, you're going to take 30 mils of that and spin it at high speed, 12,000 times G for 10 minutes. And I'll let you figure out how to do that. You'll be given these little tubes. Okay, these are going to fit in the other SS34 rotor, the little one. And after done spinning them, you'll end up with a nice pellet like I have down there. You pull off the supernatant, and now the rest of this, what you're trying to do is get these things ready to electroporate. Because when you electroporate, you're going to shoot electricity onto these. And if these have salt or other debris or dirt on them, it's not going to work. So to get these things what's called competent or ready to accept DNA for electroporation, you got to wash these extensively. The good news is the procedure is super easy, barely an inconvenience. All you do is take ice cold water. I've got a bunch of this that's ice cold. We're going to go up to chemistry and steal their ice. You're just going to basically wash this pellet extensively in 1.5 mils of water, ice cold water. You do this, it says resuspend in at 1.5 mils of this cold water. Then you pellet it at high speed. Okay, we're going to put it into a little Eppendorf tube so we can use that, one of these guys, which is of course much easier to use, much handier. And you basically repeat that. It says cleaning this thing 4 times, and I'll show you some of the particulars. Okay, so a myxo pellet turns out to be really darn sticky. And so it's difficult to resuspend, unfortunately. No! You're going to have to take your 1.5 mils. I'll give you probably 5 mil pipettes. And you can do this, I used to mouth pipette. You can do this with one of these repeater pipettemen or pipetters, or with a regular like green one. They work just fine. But you're going to take your 1.5 mils of ice cold water, and when you resuspend a myxo pellet, you got to do it like we did it before, which is you got to add your water, and you got to suck up the water and then put it over the pellet. Suck it up, put it over the pellet. You do this back and forth, and the myxos, it's very sticky, so it can take you like 2 minutes or sometimes more to resuspend the pellet itself. So it's not easy to do. It's not like resuspending a pellet of E. coli. So it takes a while. But when you are ready, you suck this up, put it into a waiting clean Eppendorf tube, and then you spin this out. It says high speed, 14,000 RPM, which is the highest speed you got, for 3 minutes. So I'm not going to show that, but you just basically do the same thing you've been doing. Balance everything, put this thing in, high speed, 3 minutes. And all you're doing is washing the cells. You'll get a pellet and you'll get a bunch of water on top. And then all you'll do is take some more of your ice cold, you'll take off the supernatant, take some more of your ice cold water, put it on this little, in the pellet that you get, resuspend it. This time you can just use a nice micro pipette, up and down, spin it again, pull it out, get rid of the supernatant, put some more, another 1.5 mils or 1 mil of water in there. You do this for a total of 4 or 5 times, and you should end up with a nice clean pellet that we're going to add DNA to and electroporate with this little machine right here. Okay, which we're gonna talk about this. Oops, you see that? We're gonna talk about this in just a second, how this thing works, and show you what we're gonna do. Okay, so once this is spun down and I've washed these considerably, I've gone through, we'll pick back up and we'll show you the actual electroporation procedure. Okay, so here's an example of pulling out the pellet and washing it. I took 1 ml of my water and I very carefully put it in there, and I'm starting to up and down pipette. In fact, maybe what I would do next time is maybe add 500 microliters first, get it resuspended a little bit, and then add my other 500 of water. But the idea is to get this pellet nice and clean. And the problem is what I just did there, for instance, is spilling some cells.
Jeez, I'm an idiot.
But you gotta just pipette up and down carefully. You don't want to, as you're washing these cells with water, and that's actually pretty nasty to the cells. So you don't want to put the tip down there and mash them up. You want to be gentle with them. So this is what you've got to do 4 or 5 times. And then you end up, this is my 5th time, and I end up with now what we're going to label as electroporation competent mixo. So I'll say E, maybe I won't, E mixo. Okay. So I know that's ready to go. Put that on ice, which I don't have here right now. And then we're going to get ready to do the actual electroporation procedure, and I'll show you how that works too. What we're going to do is we are going to take 200 microliters of our cells that are washed and mix it with X microliters of DNA that you have, that you know what the concentration is, for instance, okay? My DNA concentration I just did turned out to be 50 nanograms per microliter, okay? What we want when we electroporate, it depends on the cell. We're going to shoot for, we want 500 nanograms of actual plasmid with these cells, okay? We're going to shoot for that. So that means if I've got a 50 nanogram per microliter concentration of my particular DNA, you know, how many for 500? You need 10 microliters. We take 10 microliters of our DNA. This is all going to be on ice. And we're going to put it with our 200 microliters of our cells. So we are going to grab put the little DNA in there. And we're going to take 200 microliters of our little washed electroporation ready mixos. And remember, you're doing this all on ice. Okay? For demonstration, I'm not. So here's my mixture. So we put that on ice and we let it sit for a couple minutes to let the DNA do its thing. In the meantime, this is a electroporation cuvette, and this is going to be on ice, cooling and chilling. And you notice when you look at this, it's got a little channel in the middle here where you're going to put your sample. So what you're going to do basically is, once this has set on ice with DNA and mixos for about a minute or so, you're going to take 100 microliters of this and put it into that chamber. And then we're going to take this over to the electroporation machine and actually do the electroporation. Okay? So this has been about that much time. We're going to take 100 microliters of our mixture of mixo and DNA. And you're going to pop it in this little cuvette. So you just stick this right down like so, put it in. Perfect. the top back on. Put this back on ice. And then we're going to show you how to set up the actual electroporator. Okay, so now what we're going to do is actually do this electroporation. And I have a couple things you have to be aware of to be ready to do. You're going to zap these little guys that again are on ice, okay? 100 microliters of your cells and mixed DNA are in here and they're ready to go, okay? But that's going to hurt these cells, okay? And you're throwing this plasmid in them that supposedly is going to make them kanamycin resistant. But you can't just take these then and stick them right onto a kanamycin plate because they're sick and they have to express the plasmid. So, or express the components of the plasmid. We'll talk about that in class, okay? So they won't, if you just immediately took the electroporated them and put them right onto a plate with kanamycin, that wouldn't work. They'd all die. So what you have to do is a step called recovery. So once we zap these, you need to have a flask with about, I don't know, 1.5 mils, 2 mils of our growth medium ready, okay, which we'll have ready for you. So then you're going to take these directly out once you electroporate them, and you're going to squirt them right into this medium. So this is just going to be regular growth medium with no selection. You don't want to select yet. And you should. You what?
What kind of medium is it?
It's called CTT medium, or casitone.
Does it have any selection?
Does it have any who?
Selection.
There's no selection in that, Michelle.
Who grows in that?
Mixobacteria. I won't edit that out. But this is going to be great. Oh, okay, where were we? Oh, yeah. So what you want to do is have this flask ready. And again, I use like 1.5 to 2 mils. You don't need much. Okay. You're going to electroporate this with the parameters that we have on this little sheet of paper here. And we'll show you that in a second. And you're going to quickly, if not sooner, take these cells, remove them, stick them in this little thing, and let that thing shake at 30 degrees for as long as possible. Ideally, you want it to shake for maybe 2 or 3 generations, which for these little mixos would be something like 12 hours. We're not going to do that. We're going to push the envelope. We're going to just let them shake as long as you can stand it, at least an hour. At least an hour to allow expression. And then we're going to put them on selective